Okay, thank you very much. My name is Richard Johnson. I work for a company called Oil Plus. We're a consultancy company, a scientific consultancy company for the oil and gas industry. We're part of John Crane, who are themselves part of Smiths, which are a major kind of engineering company. So one of the first questions I often get asked is, why are you a microbiologist working in the oil industry? What problems do microorganisms cause the industry? Well, they cause some quite major problems, really, starting with souring or reservoir souring. So this is the production of hydrogen sulfide by bacteria actually within the reservoir or within the production facilities. So the microorganisms it's usually by SRB, with sulfate reducing bacteria, or SRA as well, archaea, as has been mentioned. And these microorganisms take sulfate, which is in seawater, which is used for pressure maintenance in an oil reservoir, and they use the organics in the reservoir to reduce this sulfate to hydrogen sulfide. And as you can imagine, apart from kind of causing bad smells, this sulfide is highly toxic. This is actually a picture of one of my colleagues sampling from a sour pipeline. So there are really very major health and safety concerns with large amounts of hydrogen sulfide being produced in a oil reservoir or an oil production facility. As well as health and safety concerns, we also get problems with corrosion. This is an image of sulfide stress cracking. So where you've actually got embrittlement of steel, you've actually got small pores opening up within the steel, so you can get corrosion of steel, of carbon steel, by the actual microorganisms. And again, this can be a very, very costly process. And actually, the BP Trans-Alaskan pipeline, which shut down in 2006, it's thought to the cause of that is thought to be sulfide stress cracking. And this is a pipeline that supplies 8% of the USA's oil, so this can be a big problem and lead to big fines, high cleanup costs, and big environmental and health and safety implications. Because of this, oil companies have put quite a lot of money into modelling sulfide production, so you can get an idea of how much sulfide is going to be produced and when it's going to be produced. So this is an example of a model that we produced, a reservoir souring model. So in conventional oil production, you have your oil wells here, and then you may have some water injection. So the water is injected into an oil reservoir to actually push the oil out. So you've got like high sulfide being produced around the injection water wells, and you can get an idea of how much sulfide is being produced at what time, and this really gives an oil company idea of what kind of materials they need throughout the field's life and what their chemical costs are going to be. So they want to predict how much sulfide is being produced, because if they're going to have problems down the line with sulfide being produced, then they will need to use higher value materials in the construction of the process. And actually, using a model like this, one of our clients was able to save $500 million over the course of a year just by deciding and changing and having an idea of what materials they needed to use or didn't need to use. The other kind of major problem that they cause is corrosion. So this is different to corrosion by sulfide production. So microorganisms can actually do things like this to pipelines. So you see this is a water injection pipeline, and you've got very rapid pinhole corrosion at the bottom of the pipeline where all the water's coming in. And this here actually happened after being in operation for 3 months. So within 3 months, the bacteria have eaten through this pipeline. And you can see the pressure coming down. You've got about 120 bar pressure. So you've got particularly bad health and safety concerns with this. So, but even still, a lot of people don't understand the impact, and a lot of the problem is really not understanding what the microorganisms are doing in the system and what the processes are that are occurring. So you can imagine this is actually onshore, so it's relatively easy to see that you've got a problem here. But if you have a pipeline along the sea floor, then you're not going to notice something's happening until you see an oil slick on the surface of the water, which is really something that you don't want.
So Richard, this is a brand new pipe 3 months afterwards.
After 3 months afterwards is leaking, yeah. So
the engineer is still working in the company.
No, that is one of the big problems with the industry in general. People move around very often, so the person that designed this wasn't working there later. So really having the systems and that in place to make sure it's right in the first place aren't there. So as a microbiologist, it's good to have to be able to tell a company exactly what the problems are going to be, what the negatives, positives and negatives are on these very costly decisions for different metallurgies and things. So there's another example of a subsea pipeline in the North Sea which ruptured after 5 years. So this cost, as a subsea pipeline, it's cost £4 million to repair and clean up afterwards. And while that pipeline wasn't running, it cost the company £50 million in deferred production costs, and also the cost of a new pipeline, which is kind of hundreds of millions of pounds. A final good example is a plant in Kazakhstan, which was built, and after 6 months it had to be decommissioned, it had to be shut down because of microbial corrosion. So it was running for 6 months, but the whole plant initially cost $50 billion. So this cost $50 billion for that whole 6 months of operation, so it had to be shut down and rethought. So really having an idea of what the microorganisms are going to do to that plant before you build that plant is really key, I think. So I'll show you. So the same pipeline there, you can see the hole that the microorganisms have made in that pipeline. And what it is, it's not just one type of microorganism, it's often quite a complex mix of different groups of organisms. And you can see in this case, this is a model or an idea of what's happening. So you've got SRP, which are archaeobacteria, sulfate reducing ones, producing hydrogen sulfide, and this strips the iron from the steel. But then you've got another group of archaeomethannogens helping speed up this process by mopping up the extra protons and producing methane to kind of get a cycle of corrosion. So this is how you get this kind of corrosion so rapidly. And it's estimated that 1 to 5% of GDP, gross domestic product, actually goes on combating corrosion every year. And there's a report a couple of years ago by the Navy estimating that microbial corrosion accounted for about 20% of total corrosion. So from this, at the lower estimate of 1%, you get an estimate of $143.7 billion that microbial corrosion is costing the world every year. As well as causing corrosion, you also get filter plugging. So water quality has to be of a varying standard for disposal or cleanup. So you get the microorganisms growing on the filters. You also get reservoir plugging. So a lot of people think that an oil reservoir may be just a big hole in the ground, but I brought some examples of an actual oil reservoir here. So the actual oil is actually within the holes within the rock. So this is a sandstone reservoir. Yeah, I'll pass this around, but you can actually see the microorganisms, or the oil and the water, will be within that rock, and that's a limestone one as well. So if you've got microorganisms growing on the near the well region, you're going to very quickly plug a reservoir or plug a well, and it would cost roughly $100 million to drill a new well, or to put a new well in. So this is again something that you really don't want to be doing. As well, you'll also get a reduction in separation efficiency. I'll show you later or show you in a minute how an oil reservoir or oil production system works. And you'll also get a reduction in product quality. So if you've got high enough amounts of hydrogen sulfide in the oil, you can't sell it. Even if you have lower but not maximum amounts, you'll get less money for that oil. So it can be very economically unfavourable. So to give you a rough, quick idea of what's happening in an oil system, so you have your oil well here drilled into the oil phase, and the water or the oil comes out of the ground, but it's not just a nice steady flow of oil, it's a mix of water, oil and gas. So as it comes out of the ground, it's separated in essentially a big tank. Your gas and your oil are exported out. So these can have, the microorganisms can cause problems with high levels of H2S in these exports. They can also cause corrosion in the well down here. And then your water goes for produced water treatment. So it will be cleaned up either for discharge to exact standards or for water injection again to various standards. And all through this process they can cause corrosion of the equipment. And then finally, if the water is being re-injected in a new well, you can cause plugging of the new well or corrosion of the new well. So they really can eat away at everything within an oil field. So predominantly the oil or the industry takes samples or should take samples regularly. So you've got water samples which can just be taken from pipes, and another thing you can do is take solid samples. So these are what we call PIGs. They're actually, I didn't realise what they actually stood for, but they are pipeline inspection gauges. So they just use them like these on like a brush. So it just essentially gets flown through the pipeline and it collects all the crud on the outside of the pipeline. You can get solid samples of the biofilms growing on the pipeline on the outside. This is also used for cleaning up pipelines, and you can see an example of the stuff that comes out. So we can look at these. We can look at the bacteria growing in biofilms on the outside. We can also look at microorganisms growing in the fluids that are flowing through the pipeline. To give you an idea of where the industry was kind of 5 to 10 years ago, for some companies even now, the way that they check to see if they've got microorganisms or to see if they're going to have a problem with microorganisms is take a ml of water and inoculate it into a bug bottle. If it grows, they've got a bacteria. If it doesn't grow, they don't have bacteria. That's as simple. That's the general level of understanding that we're going against. And you can actually see in this case there's a nail there, and the black is actually the iron sulfide produced from the corrosion of that nail in there. So that's for SRB. This is for aerobic bacteria. So they've essentially just changed the pH, and there's a pH indicator in there, and with a reduction in pH you get a change in colour. So that's kind of where the industry has been. This is what the engineers that work there understand. So from that we've gone to now doing qPCR, both of the 16S gene and also of functional genes, to let them know what groups are in there and how many there are. So you probably well know as well now, this is all based on culture-based methods which only actually culture 10% at very best of the population that's in there. So we've got a more accurate reading now of exactly how many microorganisms are in there, what groups of microorganisms are in there, and also now being able to tell them what types of microorganisms are in there using next generation sequencing, first using Roche's 454 and now using Illumina MySeq. So really, I was going to try how this helps the engineers that are making those decisions at the minute. We've got a puzzle. We've got a puzzle with lots of pieces. We know we have all the pieces of the puzzle, but now the real challenge is putting those pieces together and getting a kind of coherent picture and making that into something that can be used to make decisions on, for example, how an oil field will be produced, or how the microorganisms will be combated within the production, how to try and stop them from essentially destroying the plant. So that kind of really brings me back to my initial question, how we're hoping that metagenomics will be able to help the oil industry. Firstly, we want to try and answer the questions, what microorganisms are in there, and how many microorganisms are in there, how big is a problem, either as a potential problem in the first place or flagged up from routine monitoring. So I think those two in green we can quite happily answer now. The first one with sequencing, the second one with qPCR. We can kind of relate this back to these questions: what are they doing and how can we stop them? But this is only based on culture data we've got from experiments going back. So we really want to know, are they doing the same things in the field as what they were doing in our culture experiments? Do these two things match up? And I think moving on, we're hoping to look at are there any trends or patterns? How interrelated are oil reservoirs or parts of a system? So if you've got contamination in one part of a system, can we trace where that contamination came from using metagenomics and using maths to look at how related those different parts are? So that in red, I think we're maybe on the verge of this, but on the verge of being able to do, and from what I've seen here today, probably already are pretty much able to do that. So I think hopefully we can come to answer some of these problems. So I'd like to leave you on a photograph here, which is I took in a field in the South China Sea, giving a very romanticised view of an oil platform. They don't always look like that, but I'll just stop there and thank you and ask if there are any questions.
That would be a view from a hotel from the honeymoon, just through the sunset.
No, it was actually on an accommodation barge, which was even worse than the platform. So you just see the edge of it there.
Well, on the questions, I think on the last question you said there's already an answer that you can barcode, but you don't have to even do sequencing. You can do mass spec, and you will be able to get a signature from different wells. And I guess that the industry that's well known. So that's how we can track whether you are using Brent oil or something else.
All right, yeah, like GCM, yeah.
I can get that. But definitely barcoding, that would be an issue. That definitely would be easy.
Yeah. A lot of companies are actually interested in. Sometimes you can have very complex reservoirs which have either been connected in periods of geological time or are connected or moved around, and they want to know if, for example, one part of a reservoir that they're currently producing from has gone sour, will the other part of the reservoir go sour, or essentially were the microorganisms there in the first place, or have we introduced them in some way from drilling or production operations?
And also, I can think of two additional questions that the industry is posing right now. One of them would be in the kind of bioremediation, how to use bacteria to fix leaks or population, and the systems biology approach. To use, this is widely in the Middle East. I was surprised to be traveling extensively over there, and they are using these technologies to identify which bacteria might be suitable to produce energy themselves.
All right, so
that's a kind of
biofuels type. Yeah, definitely.
Oh, yeah. So I think a lot of your questions are answerable, but one of the key things in doing that would be how easy is it to actually extract bacterial DNA from oil samples? This is something we work quite a lot on. So we do have things in place now or procedures in place to extract the DNA. So usually we can quite happily separate the oil out from the water, and then the bacteria themselves. We usually use the Power Water kit, which is essentially an environmental kit. We find that's good for removing inhibitors and also cleaning up the sample and getting the DNA out sufficiently. So have you generated some metagenome samples?
No, we've got a couple of projects that are running at the minute. We're kind of very much, the industry's kind of
really taking the first steps into this this kind of area. So we've had a couple of projects on. Currently, one of the projects was going to be a metagenomics project. I was saying earlier, the problem we've had with this one is that the low numbers of biomass in the actual samples, because these were from downhole samples, so we were getting kind of picograms of DNA out and about 5 to 10 bacteria per mil from those. So really, and we've probably only got about 200 mils of sample, and it cost about $1 million to get that sample. So it's not easy to get large numbers of bacteria. So yeah, well, once they've been produced, they're generally a lot easier because they've been contaminated. So they are easier. But one of the problems we have is obviously taking the samples, getting a representative sample in the first place, and then making sure that sample doesn't change from the time it was taken to when it's analysed. So if you imagine a lot of our samples are taken offshore, and they have to be kept chilled, usually helicoptered back on shore, but it depends. I mean, we work worldwide, so it could take a month or so for samples to actually reach us. So our kind of challenges are making sure that sample stays exactly the same as it was when it was originally sampled.
One last question.
Yeah, actually, this question could be to the last speaker as well. It's really how do you set the thresholds when you get for discovering a new sequence? I mean, what are the rules that you use because you don't know what's out there, right? Well, you have some idea of the qPCR, but NGS will be more sensitive than qPCR. So you'll get sequences which are, which might be 3 sequences or something. How do you know which ones to discard and which ones to keep? What's the rule? I mean, I have an idea, but I just want to know what rules you use.
I suppose for us, we're not actually particularly interested in the low abundance sequences in there, because we're interested in the sequences of the microorganisms that are there in high abundance, and that they're going to be producing or doing the main problems. So what we would usually do would be to discount the low abundance sequences, which may be 1 or 2 sequences. As a general kind of method, I suppose, we would try and make sure that the read length was quite good, because most of our stuff's been based on 16S amplicon sequencing. So we'd try and get a read length as long as possible for that, and then try and make the data the highest kind of quality as possible, I suppose. Really, we've not seen that many completely new sequences, I have to say.
Okay, good.
I don't know if that answers me.
Let's thank you again all the speakers, and